What if I told you that the most powerful weapon against aging and disease is not something you have to buy, but something you have to feed? Right now, inside your body, there is an entire universe of microscopic life working 24 hours a day. These tiny organisms influence your energy levels, your immune strength, your metabolism, even how fast your cells age. They can either protect you or silently work against you. And the difference comes down to one simple factor: what you put on your plate. For decades, we believed aging was unavoidable and disease was mostly genetic. But science is revealing something extraordinary. The speed at which you age, the strength of your immune defenses, and your vulnerability to chronic illness are deeply connected to the health of your gut bacteria. When the right microbes thrive, they reduce inflammation, repair your gut lining, strengthen immune surveillance, and create compounds that protect your DNA. When they decline, the body becomes more vulnerable to breakdown and disease. In this video, you're going to discover which specific types of foods activate these protective bacteria, how they help slow biological aging, and how simple daily choices can dramatically reshape your long-term health. This is not about trends or fads. It's about understanding the hidden system inside you that may determine how long and how well you live. Stay with me, because what you're about to learn could completely change the way you think about food, aging, and your future. The human gut contains trillions of bacteria that regulate immunity, metabolism, and inflammation, forming a dynamic ecosystem often referred to as the gut microbiome. These microorganisms are not passive passengers. They actively interact with nearly every system in the body. About 70% of the immune system resides in the gut, where immune cells constantly communicate with microbes. Beneficial bacteria help train immune cells to distinguish between harmless substances and real threats. When this relationship is balanced, the immune system responds precisely and avoids unnecessary inflammation. When disrupted, the body may experience chronic inflammation, allergies, or autoimmune issues. In terms of metabolism, gut bacteria influence how calories are extracted from food, how fats are stored, and how blood sugar is regulated. Certain microbes break down complex fibers that human enzymes cannot digest. In doing so, they produce short-chain fatty acids like butyrate, acetate, and propionate. These compounds nourish the cells lining the colon, improve insulin sensitivity, and support healthy energy use. Some bacteria even affect hormones related to hunger and satiety, subtly shaping appetite and weight regulation. Inflammation is another critical area under microbial control. A healthy gut lining acts as a barrier, preventing toxins and harmful bacteria from entering the bloodstream. Beneficial microbes strengthen this barrier by maintaining tight junctions between intestinal cells. When the microbiome becomes imbalanced, the gut lining can weaken, allowing inflammatory molecules to circulate throughout the body. This low-grade, persistent inflammation is associated with aging and many chronic diseases. The gut also communicates with distant organs, including the brain, through chemical signals, immune pathways, and the vagus nerve. Through this extensive network, gut bacteria influence mood, cognitive function, and overall resilience, making them central regulators of whole body health. Certain beneficial bacteria in the gut produce short-chain fatty acids, SCFAs, including butyrate, acetate, and propionate, through the fermentation of dietary fiber that the human body cannot digest on its own. Among these, butyrate is especially important for healthy aging. It serves as the primary energy source for the cells lining the colon, helping maintain a strong and intact gut barrier. When this barrier is healthy, it prevents harmful toxins, pathogens, and inflammatory molecules from leaking into the bloodstream, reducing systemic inflammation that accelerates aging. Butyrate also plays a direct role in regulating the immune system. It helps control the activity of immune cells, promoting balance rather than overreaction. By calming excessive immune responses, it reduces chronic low-grade inflammation, a major driver of age-related diseases such as cardiovascular disease, type 2 diabetes, and certain cancers. This anti-inflammatory effect contributes to what scientists associate with slower biological aging. Beyond inflammation control, SCFAs influence gene expression. Butyrate acts as a signaling molecule that can turn specific genes on or off through epigenetic mechanisms. These changes affect cellular repair processes, oxidative stress resistance, and even program cell death of damaged cells. By supporting proper cellular function and reducing DNA damage, butyrate helps preserve tissue integrity over time. SCFAs also improve metabolic health by enhancing insulin sensitivity and supporting balanced blood sugar levels. Stable glucose regulation reduces stress on cells and lowers the risk of metabolic disorders that are linked to accelerated aging. Additionally, these fatty acids help regulate appetite hormones, influencing body weight and energy balance. The production of SCFAs depends largely on the presence of fiber-loving bacteria and consistent intake of plant-based fibers. When fiber intake is low, beneficial SCFA-producing microbes decline, reducing these protective effects throughout the body. Gut bacteria influence biological aging by interacting with core mechanisms that determine how quickly our cells and tissues decline over time. One major pathway involves telomeres, which are protective caps at the ends of chromosomes. Each time a cell divides, telomeres shorten slightly. When they become too short, cells lose their ability to function properly or replicate. Chronic inflammation and oxidative stress accelerate telomere shortening. A balanced gut microbiome helps reduce systemic inflammation and produces antioxidant compounds that limit cellular damage, thereby helping preserve telomere length and cellular vitality. Oxidative stress is another central driver of aging. It occurs when harmful molecules called free radicals outnumber the body's antioxidant defenses, damaging DNA, proteins, and cell membranes. Certain beneficial gut bacteria produce metabolites, including short-chain fatty acids, that strengthen the body's antioxidant capacity. These compounds enhance mitochondrial function, the energy-producing structures inside cells, reducing the excessive generation of free radicals. By supporting efficient energy metabolism, the microbiome lowers cumulative cellular wear and tear. Chronic low-grade inflammation, often described as inflammaging, is closely tied to microbiome balance. When harmful bacteria dominate, they can weaken the intestinal barrier, allowing inflammatory molecules to leak into circulation. This persistent immune activation contributes to tissue breakdown, insulin resistance, cardiovascular disease, and neurodegeneration. In contrast, beneficial microbes reinforce the gut barrier and regulate immune signaling, keeping inflammatory responses proportionate and controlled. Gut bacteria also influence epigenetic patterns, which determine how genes related to repair, detoxification, and stress response are expressed. Through chemical signaling molecules, the microbiome can activate protective genes while silencing those linked to inflammatory pathways. These effects collectively shape biological age, which may differ from chronological age, demonstrating that microbial balance plays a measurable role in how the body ages at the cellular and systemic levels. A diverse microbiome refers to having a wide variety of bacterial species living in the gut, each performing specialized functions that contribute to overall health. Research consistently shows that greater microbial diversity is associated with a lower risk of chronic diseases such as heart disease, type 2 diabetes, cancer, and neurodegenerative disorders. Diversity strengthens resilience. When many beneficial species coexist, they create a balanced ecosystem that prevents harmful bacteria from dominating. This balance supports stable immune responses and reduces chronic inflammation, a key factor underlying most long-term diseases. In cardiovascular health, certain gut bacteria help regulate cholesterol metabolism and blood vessel function. Some microbes convert dietary nutrients into compounds that protect the lining of blood vessels and reduce plaque formation. A diverse microbiome also limits the overproduction of harmful metabolites that may contribute to arterial stiffness and hypertension. By influencing lipid balance and inflammatory signaling, gut diversity plays a direct role in protecting the heart. In metabolic conditions like type 2 diabetes, microbial diversity supports better blood sugar control. Beneficial bacteria improve insulin sensitivity and help regulate glucose absorption in the intestine. They also produce short-chain fatty acids that enhance metabolic flexibility, allowing the body to switch efficiently between energy sources. When diversity declines, blood sugar fluctuations and fat storage tend to increase, raising disease risk. In cancer prevention, a healthy microbiome strengthens immune surveillance. Diverse bacteria help immune cells recognize and eliminate abnormal cells before they grow uncontrolled. Some microbes produce anti-inflammatory and anti-proliferative compounds that directly inhibit tumor development. In neurodegenerative diseases, gut diversity influences the gut-brain axis. Beneficial bacteria reduce inflammatory signals that can travel to the brain and contribute to cognitive decline. They also produce neurotransmitter precursors that support brain health, demonstrating how microbial variety contributes to protection across multiple organ systems. Chronic inflammation, often described as inflammaging, is a slow, persistent activation of the immune system that develops over time and plays a central role in the aging process. Unlike acute inflammation, which is a short-term protective response to injury or infection, chronic inflammation lingers quietly in the background. It may not cause obvious symptoms at first, but it steadily damages tissues, disrupts cellular function, and accelerates biological decline. This low-grade inflammatory state is now recognized as one of the strongest predictors of age-related diseases. As the body ages, immune regulation becomes less precise. Cells that once responded efficiently to threats may remain partially activated, releasing inflammatory molecules such as cytokines into circulation. Over years and decades, this constant signaling creates wear and tear on blood vessels, organs, and even DNA. It contributes to plaque buildup in arteries, insulin resistance in metabolic tissues, and degeneration of neurons in the brain. Conditions such as heart disease, type 2 diabetes, arthritis, Alzheimer's disease, and certain cancers are closely linked to this ongoing inflammatory burden. One key driver of inflammaging is cellular senescence. As cells accumulate damage, they enter a state where they stop dividing but do not die. These senescent cells release inflammatory chemicals that affect neighboring healthy cells, amplifying tissue dysfunction. Oxidative stress further compounds the problem by generating free radicals that injure cellular structures and trigger additional immune responses. Lifestyle factors strongly influence chronic inflammation. Poor diet, excess body fat, chronic stress, physical inactivity, and disrupted sleep all elevate inflammatory markers. In contrast, nutrient-dense foods rich in fiber and antioxidants, regular physical activity, and a balanced gut microbiome help regulate immune signaling and reduce systemic inflammation. By lowering persistent inflammatory stress, the body preserves tissue function longer and slows many mechanisms that drive aging at the cellular and organ level. Fiber-rich foods play a central role in shaping the gut microbiome because dietary fiber serves as the primary fuel source for beneficial bacteria. Unlike proteins, fats, and simple carbohydrates, fiber is not digested by human enzymes in the stomach or small intestine. Instead, it travels intact to the colon, where trillions of microbes ferment it. This fermentation process produces short-chain fatty acids such as butyrate, acetate, and propionate, which support gut integrity, metabolic balance, and immune regulation. When fiber intake is high and varied, it encourages the growth of diverse bacterial species. Different types of fiber, soluble, insoluble, resistant starches, and prebiotic fibers, feed different strains of microbes. For example, soluble fibers found in oats, beans, lentils, apples, and flax seeds dissolve in water and form a gel-like substance that beneficial bacteria readily ferment. Resistant starches in foods like legumes and slightly green bananas also nourish microbes that produce protective compounds. The more variety in plant-based foods consumed, the broader the range of bacterial species that can thrive. Increased microbial diversity strengthens the gut ecosystem. A well-fed microbiome creates a competitive environment that limits the growth of harmful bacteria. It also reinforces the intestinal barrier, preventing toxins and inflammatory molecules from entering the bloodstream. As beneficial bacteria break down fiber, the short-chain fatty acids they produce reduce inflammation, enhance insulin sensitivity, and improve communication between the gut and immune system. Low fiber intake, common in highly processed diets, deprives beneficial microbes of nourishment. In response, some bacteria may begin to degrade the protective mucus lining of the gut for energy, weakening the barrier and increasing susceptibility to inflammation. Over time, this imbalance can contribute to metabolic disorders and chronic disease. Consistently consuming a wide range of vegetables, fruits, legumes, whole grains, nuts, and seeds provides the steady supply of fermentable material that beneficial microbes require to maintain resilience and promote long-term health. Fermented foods introduce live beneficial microbes into the digestive system, contributing to a more balanced and resilient gut microbiome. These foods are created through controlled microbial growth and enzymatic conversion of food components, typically involving bacteria or yeast. During fermentation, natural sugars are broken down into acids or alcohol, which preserve the food and create an environment rich in living microorganisms. When consumed regularly, certain fermented foods can help replenish and diversify beneficial bacteria in the gut. Foods such as yogurt with live cultures, kefir, kimchi, sauerkraut, miso, and traditionally fermented vegetables contain strains of lactobacillus, bifidobacterium, and other microbes that support digestive health. These organisms can temporarily colonize the gut and interact with resident bacteria, enhancing microbial balance. Even if some strains do not permanently establish themselves, their presence can stimulate positive shifts in the existing microbial community and support beneficial metabolic activity. Fermented foods also strengthen the intestinal barrier. Beneficial microbes produce compounds that enhance tight junctions between intestinal cells, reducing intestinal permeability. A stronger barrier prevents harmful bacteria, toxins, and inflammatory molecules from entering the bloodstream. This protection lowers systemic inflammation and supports immune stability. Many fermented foods additionally contain bioactive peptides, vitamins such as B vitamins and vitamin K2, and organic acids that further promote metabolic and immune function. The fermentation process can also improve nutrient availability. It partially breaks down complex compounds, making certain minerals and vitamins easier to absorb. In some cases, fermentation reduces anti-nutrients and enhances digestibility, especially for individuals sensitive to lactose or certain plant components. Regular intake of fermented foods has been associated with improved immune responses, reduced inflammatory markers, and greater microbial diversity. By supplying beneficial microbes and supportive metabolites, these foods help maintain a balanced gut ecosystem that contributes to long-term resilience and disease resistance. Polyphenol-rich foods such as berries, green tea, dark chocolate, olives, and colorful vegetables contain natural plant compounds that interact closely with the gut microbiome. Polyphenols are bioactive molecules responsible for many of the antioxidant and anti-inflammatory properties found in plant-based diets. While a portion of these compounds is absorbed in the small intestine, a significant amount travels to the colon, where gut bacteria metabolize them into smaller, more active forms. This interaction creates a mutually beneficial relationship between dietary plants and beneficial microbes. Certain gut bacteria use polyphenols as substrates for growth, meaning these compounds selectively nourish protective microbial strains. As these bacteria break down polyphenols, they produce metabolites that enhance antioxidant defenses and regulate immune signaling. This process supports microbial diversity and helps beneficial species compete against harmful bacteria. Over time, regular intake of polyphenol-rich foods can shift the microbial balance toward a more resilient and anti-inflammatory profile. The antioxidant properties of polyphenol metabolites reduce oxidative stress, which is a major contributor to cellular aging and chronic disease. By neutralizing free radicals and supporting mitochondrial efficiency, these compounds protect DNA, proteins, and cell membranes from damage. Additionally, polyphenols influence inflammatory pathways by modulating the production of cytokines and other immune mediators. This reduces chronic low-grade inflammation linked to cardiovascular disease, metabolic disorders, and neurodegeneration. Polyphenols also support vascular health by improving blood vessel function and promoting nitric oxide production, which enhances circulation. Through the gut-brain axis, polyphenol metabolites may influence cognitive function and mood by affecting neural signaling and reducing neuroinflammation. The diversity of polyphenols increases with the variety of plant foods consumed. Different fruits, vegetables, teas, and cocoa products contain distinct polyphenolic profiles that nourish different microbial strains. Regular consumption of a wide spectrum of colorful plant foods helps sustain beneficial bacteria and amplifies the protective effects generated through their metabolic activity. Prebiotics are specific types of dietary fibers and naturally occurring compounds that selectively feed beneficial gut bacteria, helping them grow and function more effectively. Unlike probiotics, which are live microorganisms consumed through certain foods or supplements, prebiotics are the nourishment that fuels helpful microbes already living in the digestive tract. Many prebiotics belong to a group of fermentable fibers such as inulin, fructooligosaccharides, FOS, and galactoligosaccharides, GOS, which resist digestion in the upper gastrointestinal tract and reach the colon intact. Foods like garlic, onions, leeks, asparagus, Jerusalem artichokes, chicory root, and slightly green bananas are rich sources of these prebiotic fibers. When beneficial bacteria ferment these compounds, they produce short-chain fatty acids such as butyrate, acetate, and propionate. These fatty acids strengthen the intestinal lining, regulate immune activity, and help maintain balanced inflammation levels throughout the body. The fermentation process also lowers the pH of the colon, creating an environment that discourages harmful bacteria from thriving. Prebiotics play an important role in supporting microbial diversity. By selectively stimulating the growth of strains such as bifidobacteria and lactobacilli, they help maintain a balanced ecosystem where beneficial microbes can outcompete potentially harmful species. This balance contributes to improved digestion, better nutrient absorption, and enhanced protection against pathogens. Beyond gut health, prebiotics influence metabolic regulation. The short-chain fatty acids produced during fermentation improve insulin sensitivity and help regulate blood sugar levels. They also interact with hormones involved in appetite control, potentially supporting healthy weight management. Additionally, the immune-modulating effects of prebiotic-fed bacteria reduce chronic inflammation, which is linked to aging and many chronic diseases. Consistent intake of prebiotic-rich foods supports a stable and resilient microbiome. Because different bacterial strains respond to different fibers, consuming a variety of plant-based foods ensures that multiple beneficial species receive the nutrients they need to maintain optimal function and systemic health. The gut microbiome communicates with the brain through a complex network known as the gut-brain axis, a bidirectional system linking the digestive tract and the central nervous system. This communication occurs through multiple pathways, including the vagus nerve, immune signaling, hormone release, and microbial metabolites. Gut bacteria produce chemical messengers that influence brain function, while the brain in turn affects digestion, motility, and microbial balance through stress responses and neural regulation. One of the most significant communication routes is the vagus nerve, which acts as a direct highway between the gut and the brain. Certain beneficial bacteria can stimulate this nerve, sending signals that influence mood, stress resilience, and cognitive processes. At the same time, microbial metabolites, such as short-chain fatty acids, help regulate inflammation, which plays a major role in brain health. Chronic inflammation originating in the gut can travel through circulation and contribute to neuroinflammation, a factor associated with depression, anxiety, and neurodegenerative diseases. Gut bacteria also influence the production and availability of neurotransmitters. Many microbes are involved in the synthesis or regulation of serotonin, dopamine, and gamma-aminobutyric acid, GABA, which affect mood, motivation, and relaxation. Although much of the body's serotonin is produced in the gut, its regulation has downstream effects on brain chemistry and emotional balance. An imbalanced microbiome may disrupt these signaling pathways, potentially influencing mental health outcomes. Stress further illustrates this two-way interaction. Psychological stress can alter gut motility, increase intestinal permeability, and shift microbial composition. These changes may amplify inflammation and alter neurotransmitter signaling, creating a feedback loop between emotional state and gut health. A balanced and diverse microbiome supports stable communication along the gut-brain axis by regulating immune responses, maintaining barrier integrity, and producing beneficial metabolites. This intricate relationship demonstrates that cognitive function, mood stability, and neurological resilience are closely connected to microbial health within the digestive system. A healthy gut strengthens immune surveillance by supporting the body's ability to detect and eliminate abnormal or potentially cancerous cells before they can grow and spread. Immune surveillance is a continuous process in which immune cells patrol tissues, identify irregular cellular behavior, and trigger targeted responses. Because a large portion of the immune system resides in the gut-associated lymphoid tissue, the condition of the microbiome directly influences how effectively this protective system operates. Beneficial gut bacteria interact with immune cells, such as T cells, dendritic cells, and natural killer cells. Through constant signaling, these microbes help train immune cells to respond appropriately to threats while avoiding excessive inflammation. Certain bacterial metabolites, particularly short-chain fatty acids like butyrate, enhance the activity and regulation of cytotoxic T cells that are responsible for identifying and destroying abnormal cells. This helps maintain a balanced immune response that is both vigilant and controlled. A strong intestinal barrier is also essential for effective immune surveillance. Beneficial microbes reinforce tight junctions between intestinal cells, preventing toxins and harmful bacteria from leaking into the bloodstream. When the gut barrier is compromised, chronic inflammation can develop, weakening immune precision and diverting resources away from detecting emerging abnormal cells. Persistent inflammation may also create an environment that encourages tumor development by promoting DNA damage and abnormal cell signaling. The microbiome additionally influences how the body responds to treatments. Research has shown that certain gut bacteria enhance the effectiveness of some immunotherapies by improving T-cell activation and overall immune responsiveness. This demonstrates how microbial balance affects not only prevention, but also the body's ability to fight disease when it arises. Microbial diversity contributes to a broader range of immune interactions, increasing adaptability and resilience. By regulating inflammatory pathways, strengthening the gut barrier, and optimizing immune cell function, a balanced microbiome plays a critical role in maintaining vigilant defense mechanisms against abnormal cellular growth. Daily dietary habits can reshape the gut microbiome within weeks because microbial communities respond rapidly to changes in available nutrients. The bacteria living in the digestive tract rely on what is consistently consumed, and shifts in food choices alter which species thrive and which decline. When fiber-rich plant foods, legumes, fruits, vegetables, nuts, seeds, and whole grains are introduced regularly, bacteria capable of fermenting these fibers begin to multiply. As they expand, they increase the production of beneficial metabolites such as short-chain fatty acids that support gut integrity, immune balance, and metabolic health. Studies comparing plant-based diets to highly processed, low-fiber diets show measurable microbial changes in as little as a few days. Increased intake of diverse plant fibers promotes greater microbial diversity, which is associated with resilience and disease resistance. In contrast, diets high in refined sugars, unhealthy fats, and ultra-processed ingredients can quickly reduce beneficial strains and encourage the growth of bacteria associated with inflammation. These shifts can influence digestive comfort, blood sugar regulation, and immune signaling within a short period. Fermented foods can also produce noticeable effects. Introducing yogurt with live cultures, kefir, or fermented vegetables may temporarily increase beneficial microbial populations and stimulate positive changes in the existing ecosystem. Even if some introduced microbes do not permanently colonize the gut, their metabolic activity and interaction with resident bacteria can initiate favorable adjustments. Consistency is key. Occasional healthy meals have limited impact, but sustained dietary patterns provide a stable fuel supply that shapes long-term microbial composition. Because microbes reproduce quickly, they adapt to recurring food sources, reinforcing either beneficial or harmful patterns. This responsiveness demonstrates that the microbiome is dynamic rather than fixed. Through deliberate daily choices, individuals can actively influence microbial balance, supporting improved metabolic function, reduced inflammation, and stronger immune regulation over a relatively short time frame. In conclusion, the power to slow aging and reduce disease risk is not hidden in a distant laboratory or locked inside an expensive treatment. It lives within you, in the trillions of microbes that form your gut microbiome. When these beneficial bacteria are nourished with fiber-rich plants, prebiotic foods, fermented ingredients, and polyphenol-packed fruits and teas, they produce compounds that calm inflammation, strengthen your immune defenses, protect your cells, and support metabolic balance. They influence how gracefully you age, how resilient your body remains, and how effectively you fight disease at its earliest stages. Every meal becomes a biological signal, either strengthening this internal defense system or weakening it. The remarkable truth is that your microbiome is dynamic and responsive. Within weeks, consistent dietary choices can shift your internal ecosystem toward greater diversity, stability, and protection. By choosing whole, plant-forward, minimally processed foods, you are not just eating for today. You are programming your long-term health at the microscopic level. Thank you for your time, your attention, and your commitment to learning about the science of your own body. May you move forward with knowledge, confidence, and the determination to nourish the powerful allies within you. Wishing you vibrant health, lasting energy, and the very best of luck on your journey toward a longer, stronger life.